to be looking at autoimmunity from more of a systems biology wider lens than you might traditionally see it looked at when you read medical journals and when you see how it's dealt with in conventional rheumatology practices, etc. And there's some really exciting emerging stuff that's going on. And just to let you know, my talk today is pretty much summarized in a couple of publications that I've recently put out. One was in the Open Journal of Rheumatology and Autoimmune Disease in February, and the other was in the Townsend Letter. And they're both available on my website at drdavidbrady.com in full text if you'd like them, or at the websites of those actual publications as well. There's really no doubt that there's an epidemic rise in autoimmunity and immune dysregulatory disorders. If you look at the data just from about 1950 on to the turn of the century, you see just an incredible increase in incidence of the full spectrum of autoimmune disorders, whether they're Th1 dominant, Th2 dominant, classically thought of at least, atopic disorders, they're just all going through the roof. And when you look at the epidemiological data on it, it almost looks like an infectious outbreak. That's how steep the rise is. And we're going to talk about how that may actually be the case in some cases at least, because we're going to explore a lot this morning some of the potential environmental triggers that make the immune system go awry and make the body start effectively attacking itself. And one of the things we're going to look at are different microbes and stealth pathogens and what role they may play in triggering this autoimmune epidemic. But in order to really explore this topic, we have to widen the lens in the way we look at disease in general and autoimmune disease specifically. And this really fits with the integrative, functional, sort of systems biology way that we tend to look at things, but many of our more conventional colleagues don't necessarily look at it that way. But I think the landscape is changing. You know, we're all trained in medical school, we take immunology, and when we read medical journals, this is what autoimmunity is reduced to, right? All this, you know, what's going on at the micro level with the immune cells and the cytokine patterns and everything else, and that's all extremely interesting, very valuable. It helps us figure out at a deeper level what's going on as far as the pathophysiology and coming up with potential leverage points for therapies, etc. But it really doesn't help us as clinicians in our office the next day. Right? We need to really get down to some of the foundational nuts and bolts issues and leverage points therapeutically that we can actually affect change in these people's conditions and lives. And we're going to look at some of those foundational elements that seem to be common across the spectrum of autoimmunity and not necessarily specific to any one autoimmune diagnosis. And mucosal immunity is really where the foundations are laid for how your immune system interacts with its environment. And the mucosal immunity, of course, is anywhere there's a mucosal lining that we meet our environment. And of course, the biggest place that occurs is through the gastrointestinal tract. So the GI mucosa will be concentrated on a lot in this conference, and Dr. Fasano will talk at more length about it than I will. But everyone pretty much knows that about 75% of the immune resources of the body is dedicated toward the GI tract, toward the mucosal immune response, essentially, GALT and MALT, and all these, you know, special tissues that we have there. And by the way, the gastrointestinal tract is really the only system of the body that has its own independently operating nervous system as well. That can do its basic housekeeping duties independent from the central nervous system. And of course, that's the enteric nervous system. And in that enteric nervous system is pretty much every neurotransmitter you'll find in the brain and in the central nervous system. And we know, for instance, it's often cited about 95% of total body serotonin is actually used as a messenger in the enteric nervous system. So if the gastrointestinal tract has its own independently operating nervous system, it has about 75% of the immune resources of the body. The body's not stupid. What is that saying? This is an exceedingly important place, right? The body really has to keep track of what's going on there and really finely tune the dance that it plays with all of these different potential antigenic things that it's exposed to in the gastrointestinal tract. The seeds are sown very young on the health of your gastrointestinal environment and the microbiota and your mucosal immune response, and it's affected by things that occur immediately in your life, actually, you know, in utero, and then how, whether your
you're born through natural childbirth through the birth canal? And are you seeded with all these different, organisms that you're going to have to learn to cohabitate with, or were you delivered by C-section? Were you breastfed, or were you bottle fed? All these things we're finding out have specific and very serious ramifications on mucosal immune response, on the character of your GI microbiota, even in adulthood. And once those things are set in motion, they're somewhat hard to change. They're not impossible to change, but they're not easy. Now, Mechnikov, the great Russian pathologist at the turn of the last century, the 6th ever Nobel laureate, is quoted famously with this line, "Death begins in the colon." And certainly he didn't come up with that idea all on his own. If you go back into traditional Chinese medicine, Ayurvedic medicine, European eclectic medicine, South American shamanic medicine, you can almost turn anywhere, and they've already came to that conclusion long before this time, that when the gut is bad, the organism goes bad, right? It's just, you have to have a healthy gastrointestinal environment and relationship with the food that you eat. And I think it's not very arguable at this point that in the Western societies we don't have that healthy relationship, and we don't generally have healthy gastrointestinal systems, and I'll show you some of that data. Mechnikov, of course, is the father of what is known as the orthobiosis theory, and we know the term dysbiosis a lot now. Of course, we use it in functional medicine a lot. Dysbiosis simply meaning that you don't have a good balance and optimal population of the microbiota, or the bugs that live in your gut. Orthobiosis would be a good or favorable, harmonious relationship with your microbiota. And he's really one of the modern advocates of probiotic therapy, for instance. We've had the Human Microbiome Project going on for quite a long time, and now it's actually been around longer than most of us think, but this explosion in the ability through PCR technology and molecular techniques to actually dive down, do a deep dive and look at not only us, but all of the microbes that live with us in hopefully a harmonious existence, but looking at us not just as a organism with our own biochemistry and physiology, but looking at humans through a superorganismal context, meaning we're not just us, we're the sum of us, our biochemistry and metabolism, and all of the bugs that live in us and on us and their biochemistry and metabolism. It's really, what does it all sum up to, and what does it produce in the end? And that's what the Human Microbiome Project is helping us unravel some of those mysteries, including looking at, in very detailed ways, at the microbiota, or the composition of the bugs that live in our intestinal environment, whether they're the friendly, probiotics, whether they're pathogens, whether they're opportunistic organisms. We can start looking at different patterns, because in the intestinal lumen that runs right through the middle of us, but really is not within us, it's technically exterior to us, it's just littered with antigenic material. There's tons of microbes of various types, bacteria, viruses, fungi, parasites, right? And then there's all this food that we throw down it every day with all these proteins that, if they're not digested properly, if they're not prepared properly before they're exposed to the immune system proper, they're exceedingly antigenic and can cause an immune storm, essentially. And we're really guarded by a one cell lining thick mucosal lining, right? The GI mucosa. And if that thing is not healthy and doing its job, and its job is a very difficult one. It has to be a traffic cop, as we know. It has to be very efficient at bringing through things that we need from our food mass, but it has to be equally as efficient as keeping things out that might be antigenic or overwhelm us from a toxicology standpoint. So as we interface with all these commensal bacteria and all these, these potential food antigens, the traffic cop is right here, and if the mucosal health degrades and you get hyperpermeability or so-called leaky gut, it really opens up the faucets for exposure, not only at the mucosal immune level, but down deeper in a more robust immune response, which is more specific to the patient genetically based on how they present certain antigens, which is what brings us, genetic susceptibility to these sensitivities and autoimmune diseases, which we'll explore a little bit more as we go. But suffice it to say that it's very, very important that this barrier is very, very healthy. Let me give you a little example here. This is from Nature in 2008. A microbial symbiosis factor prevents intestinal inflammatory disease. And what they did in this study is they used a human symbiont, Bacteroides fragilis, and showed that it protected animals from experimental colitis induced by another bacteria, Helicobacter hepaticus. And what they did here is they inoculated wild-type mice with the Bacterium H. hepaticus and created essentially an experimental mouse version of inflammatory bowel disease, which is an autoimmune disease. When they introduced the H. hepaticus into the microbiota of these animals, it activated Th17 cells, which then release a series of cytokines which are pro-inflammatory, like IL-17, for instance, which causes the symptoms of this inflammatory bowel condition. But then they introduced B. fragilis, which expresses a certain polysaccharide A, and they added that to the gut. The dendritic cells were able to take up that PSA on their surface and activate CD4 T cells and regulatory T cells, which in turn release IL-10, which is the enemy of IL-17. So it alleviated the disease. So why do I tell you this? And quote from the literature, which I don't really particularly like to do when I'm speaking, but it's worth it in this case. Because what they did is they took animals that were healthy, they introduced one bacteria into their microbiota, and they induced autoimmune inflammatory bowel disease. And then they eradicated it by introducing one more bacteria. That's pretty powerful, right? It tells you that, even a slight shift in the microbiota can have very large effects. Now, if we look at disorders that are associated with dysbiosis or changes in the microbiota, and look at disorders that are also associated with intestinal hyperpermeability or leaky gut, the list is very, very long. And this is not complete. This is just a few of them I pulled out that you can pull out of the literature. But you see, it's all your itises, right? All these inflammatory disorders, but a lot of what we would consider autoimmune or immune dysregulatory disorders. Anybody see any of that stuff in their office, right? I mean, all day long. And that's the stuff that's growing at epidemic proportions. But these changes, these associations or patterns of dysbiosis, specific to many times the genus or species of the organism that's out of whack, is not just unique to what we would consider autoimmune diseases. For instance, in autism, there's been a lot of work done mapping the microbiota in children with autistic spectrum disorder. And what you see very clearly is versus controls, autistic spectrum subjects have much higher levels of Clostridium in their GI microbiota. And they cluster it out into different groups of organisms. But it's very clear that the microbiota or the GI environment is very different in autistic spectrum subjects than those who don't have autistic spectrum disorder. Now, does that mean that it's the Clostridia or the change in the microbiota that's causing all autism? No. I definitely don't think that that's the case. It's part of the puzzle, right? And then you get into the argument, well, which came first? Did the autism shift to microbiota, or did the shift in microbiota contribute to the autism? And we're just really starting to unravel that, and you see the researchers really looking beyond these associations and now looking into causality, and they're actually showing causality in many different cases. And I think I have a slide in here showing you something that's fairly new, really showing causality in overgrowth of certain organisms in autoimmune joint disorders like rheumatoid arthritis. But if this is true, if the microbiota is very important in how we assess people that have autoimmune disease or have a strong family history of autoimmune disease, how do we map the microbiota as clinicians, right? We don't have research labs, and we know that the traditional techniques that have been out there using microbial plating and growing out organisms, with the sample collected in a nutritive broth and sent to a lab, and they grow it out and they do a comparative sugars test and they try to figure out what's there, they're extremely limited in what they can do because you're only basically seeing the anaerobes and some of the facultative organisms, which make up about less than 5% of the total microbiota. But with new molecular techniques, we can look way beyond that. So now with PCR, where they can take a biological, you can take a biological sample. You have a patient collect, you know, a sample of stool, and instead of dropping it in a nutritive media where the bacteria and other organisms are still growing because there's sugars in there to keep them alive till they get to the lab and they plate them out, right? Instead, it's dropped in formalin, so it kind of freezes everything on where it was when it came out of the patient. Because when you're using PCR and molecular techniques to hunt the DNA of these organisms, they don't have to be viable. They don't have to be alive. You don't have to plate them. You don't have to grow them. So even if they're dead, you know, you've dropped them in formalin, you find the DNA. And if you find the DNA, the organism was there. So you can not only identify the organism, but then you can use a methodology that you can quantify it, basically. You can determine how much of it was there. So using PCR and molecular techniques now, it's really revolutionized our ability to study these things and to create these associations and then start looking for evidence of causality. There's some inherent advantages of using molecular or PCR DNA-based techniques over traditional culture techniques. One of them I already mentioned. You can now identify the majority of the bugs, which are the anaerobes, in addition to the aerobes. So you can look at essentially the whole microbiota if you put in all the probes for those organisms. You can eliminate the errors in growth during transport. Like I said, in the old techniques, you know, it's a dirty little secret that we don't really want to acknowledge as clinicians, but you have the patient, you know, collect the stool sample, they put it in the nutritive broth, and the balance of organisms that was in that stool sample when they dropped it in that nutritive media, by the time 3 days later it's at the lab and they're plating it out, it's entirely different. Because some of the organisms loved the sugars in the tube and others didn't. So some flourished, some didn't, right? Some were suppressed. Some loved it so much they flourished, and you'll see the, like, if you look at, for instance, some of the yeast in that sugar medium, they'll just go through the roof, and then after about a day or two, they'll burn out and then drop down. So it's all over the map when you look at the studies of that. But if you look at some of the differences in sensitivity of the molecular techniques, it requires only about 1 to 5 cells for identification of bacteria versus about 1,000 to 5,000 for culture, for colony-forming units to form. With parasites, it's about 5 cells per gram for DNA detection versus about 25,000 cells per gram with standard techniques. So that's a major landscape change. And the other thing is you don't have to collect the sample 3 times, you know. Remember the old O and P times 3, right? Well, I think you guys all know this. If you have a patient who likes you a lot and you have them do a stool collection, right? After they collect the stool once, they like you a little less. After you have them do it 3 times, they don't like you much at all, right? So if you could just have them collect 1 sample, it's an inherent advantage. So why would it be maybe very powerful to be able to really get a handle on what their microbiota looks like? Well, if you look at, and this is once again just a very partial list, at some of the examples of epidemiological associations between overgrowth of different GI microbes in the GI tract of subjects who have specific autoimmune diseases versus not, you see definite patterns. And once again, I want to caution that I'm not suggesting that every one of these associations guarantees causality in the disorder, but I think they're very much in play in the causality of these disorders, and I'll show you the reason I think that in a little bit. But if you look, for instance, at people with inflammatory arthropathies, whether it's something like ankylosing spondylitis or rheumatoid arthritis, you see overgrowth of Klebsiella species, of Citrobacter species in the GI tract. You see Proteus overgrowth in the urinary tract in rheumatoid arthritis. I'll show you some studies on that. You see overgrowth of Yersinia enterocolitica in autoimmune thyroiditis. Overgrowth of E. coli and Proteus just in general in autoimmune disease, Campylobacter jejuni and Guillain-Barré syndrome. So there's many, many more associations along the way. And what you really have going on here is if you have certain individuals who may have a genetic predisposition or an environment in the GI tract that's conducive to overgrowth of a certain, let's say, microbe, for sake of argument, let's say a bacteria, and these bacteria express certain peptide motifs on their surface, the immune system uses that to try to combat that organism, right? It builds antibodies against that antigen specifically. If that antigen has a structure or an amino acid sequence or a motif that is similar enough to a protein structure on your host tissue, and you're already inflamed, it doesn't take much for it to not discriminate anymore and cross over. So let's say a subject had overgrowth of, let's say, Klebsiella or Citrobacter in their gut for some reason. Their body's reacting to try to dampen down that GI infection by creating antibodies to bind to these antigens on this organism, and you get antigen-antibody complex formation. But you have some peptides expressed on the synovial linings that are very similar to those on Citrobacter and Klebsiella. You can have a crossover reaction, and of course this is the classic molecular mimicry pattern, right, where some environmental antigen that you're exposed to is close enough structurally to some host structure, and one structure mimics the other enough that the immune system stops discriminating between the two. And this is, you know, there's evidence for this across a very wide spectrum of autoimmune disorders. It's not unique to any one. And if you want to look more into some of the nuances or the more specific mechanisms of this, this was a great paper in Mucosal Immunology in September of 2008 called Joint Expedition Linking Inflammation to Arthritis. Really interesting stuff. And if you look beyond that, especially if you move beyond sort of the literature in the United States and in some of the larger Western countries, this, for instance, is something out of the Ukraine, but it says the colonization resistance of mucous membrane of large intestines in patients with rheumatoid arthritis in periods of exacerbation. What they did is they mapped the microbiota in normal subjects and subjects with rheumatoid arthritis, and they followed the changes in the microbiota in RA subjects as their disease either went into quiescent periods or exacerbated. Now, one thing that we know is fairly common in many autoimmune diseases is they wax and wane, right? That you'll have someone who really flares up with RA or ulcerative colitis or MS. They're really in a bad, you know, jag, and then all of a sudden they'll just sort of spontaneously kind of get better. And they feel a lot better for a while, and then they know it's coming again, right? So the disease does this. And the question has always been, why? What's doing that? Is there an infectious agent, a microbe? You know, what is going on? Well, when they looked at the microbiota here in healthy people, they found, you know, a fairly expected population of different microbes, bifidobacteria, lactobacilli, Bacteroides, and so forth. When they looked at RA subjects who were symptomatic, they found a very different looking microbiota full of enteropathogenic organisms, Citrobacter, Klebsiella, and so forth. But what they then found out is when the patients with RA went into a relatively quiet period and they were asymptomatic, their microbiota composition looked like normal people again. And when they exacerbated, it had this specific aberrant pattern. So once again, what comes first? The shift in the microbiota brings the disease to express itself and be symptomatic, or is it the other way around? I don't quite know yet, but they really took an incredible leap at the end of this paper when, if you read it here, it says, taking into account significant changes of colonization resistance in the colon mucous membrane in remission periods of RA, it's necessary to apply bacteriotherapy using bacterial drugs containing bifidobacteria and lactobacteria. All I can take from that essentially is that if you call them bacterial drugs instead of probiotics, they work better. Anyway, what you have going on here is you're seeing, as they're starting to look at the microbiota, that you can almost dictate the shift in how the disease behaves by changes in the microbiota, which is really, really interesting stuff. Right? So not only can these patterns help us potentially diagnose people or even find people who may be at risk for developing it later, and if we can do that, maybe we can intervene and we can slow down the train, right? We can sort of short-circuit the pathophysiological process so they don't really develop that disorder. And if you look at this paper here, it's just another one talking about these relationships that they found increased immune responses to Klebsiella in patients with ankylosing spondylitis, ulcerative colitis, Crohn's disease, and in Proteus in patients with RA, and on and on. And if you start really doing molecular-based stool analysis on these patients, either with autoimmune disease diagnoses, previously. Those who you suspect have it from clinical exam and other laboratory studies. But even those with strong family histories of autoimmunity, you start seeing these patterns. This, for instance, is a patient of mine who had early inflammatory joint disease, and you can see in the opportunistic bacterial section here, they had increased DNA recovery of Citrobacter furundi and a Klebsiella species. So if you can go ahead and eradicate that, will you stop their disease? Will you make any joint damage that they might have suffered from it go away? You know, you're not going to fix the damage that's done, but you may slow the process if that's part of, you know, the dominoes that are falling that's dysregulating the immune system and causing this molecular mimicry, kind of fueling the fire, if you will. And, you know, these concepts aren't entirely brand new. I put this in there just to show you that it's been in the literature for a while. This is the Journal of Rheumatology back in 1990, reflections on the link between intestinal permeability and inflammatory joint disease. And in this paper, this was actually a commentary in reaction to a paper showing that, you know, in 70% of the cases, they were able to strongly confirm that, you know, there was significant inflammatory changes in the gut and in hyperpermeability in inflammatory joint disease. So they go on to say joint disease is triggered through the gut. This is back in 1990. It's a pretty long time ago now, but really the day in, day out practice of rheumatology, when a patient goes to the rheumatologist and they get diagnosed with rheumatoid arthritis or some other inflammatory joint disease, do they get stool analysis? Do they get any kind of attention paid to their gastrointestinal health? Generally, the answer is no, right? If they did, it would be a referral to a gastroenterologist. You know, they scope them this way, scope them that way, and if they don't find anything overt, then, you know, purple pill, right? I mean, it's really not the kind of, you know, comprehensive care we need from a systems biology standpoint to try to slow down this train. And, you know, I've been talking about the GI microbiota and stool analysis and looking at things down, you know, really distally in the GI tract, but we need to remember that the GI tract starts in the mouth. And if you look in the literature, sure enough, you see things like the incidence of oral infections and gum health, dentition having a big role to play. We know that it's linked to inflammatory risk factors in cardiovascular disease and other things that's been known for a long time. But in this case, it says the findings support the hypothesis that oral infections play a role in rheumatoid arthritis pathogenesis. In fact, P. gingivalis, one of the most common bugs to cause periodontal disease, impacts citrullination of peptides, and that's, of course, anti-cyclic citrullinate peptide antibody is one of the things we look for to see if someone is seropositive for rheumatoid arthritis. And we know that the bugs, if you have bad oral health and you have essentially dysbiosis of the mouth, if you will, you probably have leaky mouth as well. And these things spread. They spread around. This is why you have increase in incidence of cardiovascular disease in these types of situations. So, leaky gut is not only in the, in the large intestines and small intestines. It's all the way up to the mouth. And here's another paper looking at another organism that's involved in periodontal disease, Fusobacteria. They predispose humans to colon cancer. We know that, but why? Turns out that it's probably because they cause an inflammatory bowel type of condition. They're associated with ulcerative colitis, Crohn's disease, and others, which by the virtue of those pathologies being present, you have an increase in colon cancer, right? So maybe it's just further down the line as the dominoes are falling. But this is some really interesting work. This is by Alan Emriger, and Alan Emriger has done great work in the UK at King's College, looking at some of these relationships and molecular mimicry patterns. In the case of Dr. Emriger, it's Proteus mirabilis infection, stealth infection of Proteus mirabilis in the urinary tract, and rheumatoid arthritis. And I just put a couple of his papers up here. Rheumatoid arthritis, proposal for the use of antimicrobial therapy in early cases, and rheumatoid arthritis and autoimmune disease triggered by Proteus urinary tract infections. And Emriger, I said, has done his work at King's College in the UK, but his findings have been repeated globally, essentially, by many, many different research groups. So this is not a phenomena that's local to, or regional to the UK, to that population, etc. But what he's talked about is that if you have a susceptibility to harbor a stealth infection and just not be able to beat down an infection of Proteus mirabilis in the urinary tract. It's subclinical. It doesn't have the classic signs and symptoms of, you know, what we learned in medical school as a urinary tract infection. There's no burning and urgency and all that kind of stuff. All the normal UA kind of stuff is negative. But they have this stealth infection with Proteus. And our bodies are trying to combat that by building antibodies to the hemolysin and urease enzymes expressed by the organism. And they have a structural similarity to the HLA-DR4 and some of the hyaline cartilage in these individuals, and you can get a molecular mimicry crossover that damages the synovium. And he's worked out the structural similarities between the Proteus mirabilis hemolysin and urease and these different structures. So it really kind of answers some questions that we may have had when we first learned about these disorders. Like when you learned about rheumatoid arthritis, we learned that it really only affects certain joints, the small joints of the hands, and spares other joints, right? That was a nice factoid we learned, but no one ever really told us why, right? Why does it spare all these other joints that other disorders that, you know, seem to attack, and it only hits certain joints? Well, Emringer may indeed have explained this, as well as some of the other things we know about RA, like the female predominance of 3 to 1. Well, we know that UTIs are much more prominent in females just because of the differences in anatomy. Disease onset between 30 and 50 years, this is when UTIs, chronic UTI incidence goes way up among middle and older age women. Exacerbation after pregnancy. That's common with UTIs as well, particularly after natural childbirth and some of the anatomical changes that can occur. Low concordance rate in identical twins and fluctuating course of the disease tends to suggest some level of non-genetic involvement or environmental factor in the pathogenesis of the disease. The presence of rheumatoid factor in high proportions of RA patients is likely a secondary phenomena due to B-cell stimulation due to the antigen-antibody complexes to Proteus. And then this is the lockdown kind of element of it. You see the amino acid motifs of similarity between the Proteus hemolysin and the HLA-DR molecules that are in only genetically susceptible, you know, people who are genetically susceptible to RA with that HLA pattern. And then the Proteus urease structure to type 11 collagen. Where do you find type 11 collagen? Only in the joints affected by RA. Coinky dink? Interesting. Not in the joints spared by RA. Isn't that cool? I think that's really, really cool stuff. So, you know, he has done outcome studies using not just the sort of the traditional approach to rheumatoid arthritis, you know, anti-inflammatories, moving up to steroidals, and then to even, you know, immune suppressants and biologics and chemotherapeutic agents and things like that. But really, if you look at his protocol in seropositive individuals, he's going right in with antibiotics using Cipro, and he's developed an assay that's sensitive enough to find the Proteus mirabilis in the urine, and it's a research test. There's no commercial test available right now for clinicians because docs aren't doing the test, you know. Maybe that would change if it became sort of a common, understood, accepted component of working up and treating this disorder. And then his diet is basically, you know, herbal diets. What does that mean? Cranberry juice, things high in D-mannose that are blocking agents that we know are effective in urinary tract infections because they block adhesion of these organisms to the urinary tract wall and the ureters and so forth, and the bladder wall. High fluid intake, basically to diurese the patient, urine acidification with very high dose vitamin C, and then he gets into NSAIDs and DMARDs, immunosuppressives as needed for symptomatic control. Now, one of the criticisms of this concept I'm talking about of molecular mimicry, if you read the literature, is basically that it's not universal. In other words, every person with RA would not be shown by Emringer or anyone else to have Proteus mirabilis infection in the urinary tract. Not every person with Graves' disease, Hashimoto's, would have Yersinia overgrowth. That seems to be because there's not one universal potential trigger. There's a multitude of potential triggers. And sometimes it may even require a perfect storm of multiple triggers ganging up on someone at once who has the right presenting antigens, who present it just right to their immune system, and down the track goes the train, right? In other individuals, it never gets out of the station, and they never have the expression of autoimmune disease. But this paper I found fascinating because it may actually, you know, put down in language in a study what's going on here. This is called, the title is T-cell mediated autoimmune disease due to low affinity cross-reactivity to common microbial peptides, and that's the key, common microbial peptides. The author says, we show that a microbial peptide, so a protein expressed on a microbe that you might have overgrowing, common to several major classes of bacteria, can induce MS-like disease in humanized mice by cross-reacting with T-cell receptors that also recognize a peptide from myelin basic protein, a candidate MS autoantigen. So what they're saying here is there's microbial peptides expressed by not only one single organism, but might be conserved across a whole class of organisms that has structural similarity, in the case of MS here, to myelin basic protein. So that, you know, this is the connection here. Structural analysis demonstrates this cross-reactivity is due to structural mimicry of a binding hotspot shared by self and microbial antigens rather than to some dysregulation in TCR recognition. But this is the takeaway point. These data suggest a possible explanation for the difficulty in incriminating individual infections in the development of MS or any other autoimmune disease. It's not limited to one. So it doesn't fit really nicely in the box of reductionist thinking. Everyone get it? All right, how about, I mentioned thyroid a little bit. Let's talk about specifically thyroid and different connections in this regard. Infection, thyroid disease, and autoimmunity. Molecular mimicry has long been implicated as a mechanism by which microbes can induce autoimmunity, but how about thyroid specific? This is a really good paper on viruses and thyroiditis in the Virology journal, and it talks about a much higher incidence of reactivation of chronic infections with EBV and HTLV1 and HSV and other types of very common chronic viruses in autoimmune thyroiditis. So it seems like these viral pathogens can be the environmental trigger that can induce a molecular mimicry that can create an attack against the thyroid. Another well-known one in the bacteria, Yersinia enterocolitica, which can overgrow in the gastrointestinal tract. And Yersinia on its surface shows saturable binding sites for TSH. TSH receptor antibodies then could be produced. Your body's trying to combat the Yersinia by making antibodies to latch on to this peptide that looks a whole heck of a lot like TSH receptors on the thyroid. So now you're running these things around your body trying to get them to the infection, to fight the infection in the gut, and they're passing by the thyroid and they see the TSH receptor and they go, close enough, right? Locked on, and now you just tagged your thyroid for destruction by your immune system, right? And if it's a very quick process, maybe it's Hashimoto's. If it's a slow smoldering process, it's maybe Graves', right? I haven't really worked out the nuances between those two yet. So maybe assume that the gram-negative bacterium Yersinia enterocolitica may have an active part in triggering immunogenic thyroid disease. And if you look to studies like this one in Japan, where they looked at Yersinia antibodies, you see that they're much more prevalent to be found in patients with thyroid disease in general than healthy controls. And if you start hunting around through the microbiota of these people, not only come in and say they have Hashimoto's or they have Graves', or they just have elevated thyroid antibodies when you check them, or they don't have any thyroid problems yet, but you find out their mom is on thyroid hormone because they had a problem, their aunts, or all the females are on thyroid, right? It's interesting if you start doing stool analysis on them for other things or because you're doing a complete workup on them, I find a lot of this is Yersinia enterocolitica. But once again, not the universal cause, not there every single time. In my mind, as a functional medicine doctor, I don't think I'm doing any harm by trying to make the gut as healthy as possible, by trying to restore the best microbial balance possible in the gut and the best mucosal health and the best mucosal immune response, whether it's through their relationship with food and finding which foods they are compatible with or not, whether it's using some anti-inflammatory type of compounds, whether it's using some antimicrobials for a while to try to get rid of things that we found that are out of balance and we know are associated with certain conditions. You can either, intervene with, the bigger cannons, if you will, the Cipro and the Xifaxan and, your prescription antibiotics, which tend to not be very forgiving and not sparing of the rest of the microbiota that you don't want to kill. Or you can use more strategically things like specific botanicals that have certain actives in it that we know are more functionally antimicrobial, volatile oil compounds. And if it's an organism that the lab can actually grow out and do sensitivity tests, that's one of the nice things that you can get. You can find out, based on the organism that was found, how it reacts to these different natural agents. And oftentimes they're more than enough to do the trick. So you can see here, this is just a sample natural GI, antimicrobial formulation with things like Artemisia or Chinese wormwood, standardized to the artemisinin, berberine sulfate, Berberis vulgaris, and these are all berberine containing herbs here. Arctostaphylos uva ursi, grapefruit or citrus seed extract. Caprylic acid is a great natural antimicrobial in the gut. Black walnut or Juglans nigra is a great antibacterial, antifungal, antiparasitic. And then kind of an outlier here you don't see in a lot of these formulations. This is Tribulus terrestris, standardized to the furostanol saponins. And Tribulus is used in Western herbology much more so for libido issues and trying to increase endogenous testosterone production. Quite frankly, it's not all that good at doing that. It's a much better antimicrobial in the gut, and that's how it's used in other forms of medicine, traditional Chinese medicine, Ayurvedic medicine, and the like. And it's not been used that much for that here, so it's really quite a good choice. And of course, probiotics. This is interesting stuff from Bengmark, basically saying that, a big difference between our Western cultures with autoimmunity, with poor GI health and a lot of GI disorders, and more primitive cultures, if you will, that are not really affected by modern Western diets and lifestyles. The biggest thing that they can find objectively is they have a lot more of what we consider probiotic or lactic acid producing species of organisms in their GI tract. So there's a lot of evidence that probiotics are great things, but there's a lot of controversy about them as well. And if you read a lot of work of Bengmark and other experts in probiotics, they're really way beyond the point of trying to find the magic probiotic, right? What is the strain that has the most evidence behind it? He says, stop looking for the prime violin soloist of probiotics and start using the orchestra, right? Use a wide array, a broad spectrum of high diversity of probiotics and rotate them, and it's much better. We're not going to find the magic strain. And the other thing that Bengmark talks about a lot, which I do agree with and I think is very much of a misnomer, when you look at the number of what we consider probiotic organisms and compare it to the total amount of organisms in the GI microbiota, there are way, 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 way more microbes in your gut than there are cells in your body. I mean, they outnumber us. So do they live within us, or do we kind of hang around on them? I don't know. Numerically, we definitely hang around on them. But when you give probiotics, even in that functional medicine sort of lexicon of the 4R program, anybody know that? Remove, replace, reinoculate, repair. Reinoculate, repopulate. We don't repopulate, let me tell you. When you give probiotics, you're not appreciably changing the numbers statistically in the microbiota, because it's like dropping a cupful of water in the Pacific Ocean when you give a probiotic, right? So you're not overwhelming these bad bugs. You're actually, with probiotics, you're changing the landscape immunologically more than the numbers. A little bit of probiotics go a long way. They change the cytokine expression almost immediately. In fact, even attenuated probiotics do that. Not as well, but they use attenuated probiotics a lot in like dental treatments and stuff. There's something called Dentalac. It's a totally attenuated probiotic, but yet it changes the mucosal response in the gums because of the peptides expressed by those organisms. So they're signaling molecules as much as they're anything else. And Bengmark actually describes probiotics as pixie dust, right? It's not as much how much there, it's that it is there, right? And just a little bit, it's like magic pixie dust, kind of a cool way of looking at it, but it don't get hung up on the numbers. Therefore, you know, I'm giving you a pill with 15 billion organisms. That's better than the one with 12 billion organisms. How much? I don't know. Jury's out. So Eddie, he couldn't figure out why his vegetable stand was failing. Come on, that's funny. Okay, let's talk a little bit about foods, because I've been spending most of the time on the microbes here. But autoimmune thyroid disease and celiac disease. Alessio will probably talk maybe more about this connection, but we know that overt celiac disease patients, subjects, have about 10 times the rate of autoimmune thyroiditis than non-celiacs. Coinky dink? Probably not. We know that the gluten, gliadin, sort of antigen antibody complexes have an affinity for peptides expressed on the thyroid, and you get crossover reactions and attack the thyroid. So I'm not going to spend a lot of time on testing for, whether it's overt celiac or whether it's gluten sensitivity, because the other speakers, I think, will go into that at more length. And I know Dr. Fasano's work is part of it, at least, is to trying to find new, better markers that you can actually use, not to diagnose necessarily celiac disease, but shades of gray of gluten intolerance. But there are some things we can do already. I mean, there are a lot of different tests out there, but one of the things we look at even in stool analysis is, are we seeing more, evidence of inflammation along the gut lining? Is there likely hyperpermeability and leaky gut? Is there a low level of secretory IgA being produced? Maybe because they're really stressed out, or they just genetically don't make as much. And do they, there's some markers such as the anti-gliadin secretory IgA. If you have enough IgA, and then you're producing very high levels of anti-gliadin secretory IgA at the level of the mucosa, you're right then and there, that's enough for me to pretty much take gluten out of the diet. Maybe universally anyway, but I'm just saying if a patient's giving you resistance, then say, at least here's an objective marker that you may not have a great deal of immunocompatibility with any of those substances that might have that in it. And if we look at the change in intake of our diet, changing carbohydrate intake patterns from primitive man to modern man, I mean, it's just night and day, right? Primitive man, fruits, vegetables, roots, nuts, things like that made up the virtually the entire diet, where totally different now. You're getting, 60% or so refined grains. And I think Dr. Davis will have much better information on some of this type of stuff, but we know that the diet's just massively changed in a relatively short amount of time from a evolutionary perspective. And this is a study that came out of some people, some researchers I know right up the road from me in Yale, and looking at levels of sodium chloride in the diet and how that might drive autoimmunity by inducing Th17 cells to kind of go off the rails and become extremely active. This, once again, this made the popular press as saying it's the salt that's causing autoimmune disease, right? It's always got to be the one thing, right? It's not, it's just another piece of the puzzle and another kind of confounding element in our perfect storm that we have in our modern society. If you look at this, we've known for a long time that bovine albumin peptides, cow's milk peptides, are the etiology of a lot of insulin-dependent diabetes. In genetically susceptible children who have the right HLA presenting patterns, if you give them cow's milk as a kid, like the commercials in the dairy industry tells you to do, right? And if you're not, you're a bad parent and all that, and they're not going to have any bones when they grow up and stuff like that, right? You give them dairy all along, they're building this immune response to it, and somewhere around the time they're, 11, 12, 13, 14, they hit a crossover point, And the similarity between some of those bovine albumin peptides and some of the expressed peptides on the insulin-producing cells in the pancreas, it's close enough, and your immune system takes out the insulin-producing cells. And it does it quick, right? You see these kids that are perfectly normal, and the next day they pass out and they go to the ER and they got diabetes, right? That's a molecular mimicry phenomena. But once again, milk peptides are not the only thing that have been shown to cause that same phenomena. It's been shown with viruses. It's actually been shown to some degree with insect bite venom, causing the molecular mimicry. That's the environmental antigen. All right, so what we do a lot in our practice with autoimmune patients is not only look at the microbiota and the gut health and really do everything we can to improve that, but we look at their relationship immunologically with their food. So we'll use objective cellular response tests by looking at how the white blood cell population reacts to a whole series of different foods and other substances that they may come into contact with, whether it's food, food additives, food colorings. We can even do herbs or functional foods. We can look at pharmaceuticals. There's a lot of different ways you can use this technology, which uses modified culture counter, sort of cell analyzing technology to look at the morphology, essentially. What happens to the white blood cells? Do they shrink? Do they swell? Do they lyse? Do they, degranulate? You can find that out by doing an objective, basically an electrical signature of the cells as they go through a very fine aperture right here. And that's how you do a CBC, right? It goes through a culture counter, and as the cells go through this little aperture right here, depending on how they conduct electricity or a current that's going from anode to cathode right here, then you know what kind of cell just went by. You get what's called a volume plot curve. And you can do that and get a baseline of the blood, and usually in this technique they use a lysing agent that just lyses all the red blood cells, gets them out of the picture, because they're so numerous they kind of cloud, it's like static on the line. And they're not involved in the immune response. So when you get live blood sent at the lab, they lyse all the red blood cells, they leave the white blood cells, platelets, plasma proteins, and all that intact. They'll do basically a modified culture counter analysis and get a volume plot curve of what the electronic signature of the population looks like, and then robotically it'll go pick up one food, and it'll do it 200 times or whatever. It's just isolated food proteins in the wells. It'll pick it up, do the same analysis, and then overlay the data. And it can grade any kind of shift that occurs. The only way you get a volume plot curve shift is if the cell population changed, the morphology of it. Did they break open? Did they degranulate and release, inflammatory mediators and things like that? So you can see not necessarily exactly what pathway everything occurred on, which really you don't care as a clinician. You just want to know, did this patient's immune system freak out when they were exposed to that food, or was it very tolerant of it? Okay? And that can be very powerful. Then you can modify the diet and get people off the things that they don't have good immune compatibility with. Everybody see this picture before? This just sums it up, doesn't it? This is great. Going up the stairs, going up the escalator instead of the stairs to go into the gym to get on the stair stepper, right? Insanity. I love this ad, too, from the turn of the last century, the end of the Victorian era. Fat, or eat, always stay thin. Fat banished with sanitized tapeworm. So they were inducing tapeworm infestation to compete with them for their caloric consumption so that they would get skinnier. Kind of interesting stuff. People would never stoop to something like that now, would they? And I put this slide in there just because I like to. I like to see your reaction. It's really good right after lunch when you speak. But anyway, this is roundworm infestation. These are liver flukes, and this is about a 30-foot tapeworm. And in medical school, you basically see something like this, and they say, if you see anything like this, and they just got back from Swahili or some exotic country, they have a parasite. Break out the big guns. Other than that, don't worry about it, right? That's turning out to be totally false in a multitude of ways. There are pathogenic parasites that are still very much in play and now being associated with a lot of these different disorders, including autism, by the way. I showed you the Clostridium stuff, but there's other stuff on parasites as well. But from the other side of it, that we've potentially eradicated parasites too much. This is actually a picture that was emailed to me, attached to an email on a Sunday morning by a patient, all right? After I almost choked up my breakfast, well, first of all, I didn't even want to open it. I'm like, oh no, because I know that you know the patients, you know the type, right? So you get the email on Sunday and it has the attachment. I just found this in my toilet. What is it? And you're like, all right, I'll go in, right? Boom. This comes up, Any take? I said, I don't know. I replied, I don't know, but it don't look good, right? So I did the old, try to get it, put it in a baggie, whatever. So I sent it to the hospital lab. I know the medical director, so I alerted him to the fact, this is coming down. Take a look at this. So he calls me later in the day and says, sit down. I said, is it that bad? He goes, sit down. I said, okay. I sit down. He goes, grapefruit seeds and the little things that come off them, right? I'm like, are you kidding me? He's like, no. He laughed. He says, it's happened before. So I looked like a moron, the patient looked like a moron, but it's all a good laugh. But I put it in here only for the educational lesson of you really need to think twice before you supply patients with your email address. Right? But if we look at some of the prevalence data of inflammatory bowel disease, because this is a classic autoimmune disease, right? If you look at, red is the high incidence, yellow is the medium, white is the low. Where is the autoimmune disease? In the industrialized Western countries, right? There's got to be something going on environmentally causing this because it's not in the rest of the world. And you get like South Africa and Australia. Why do they have it a little bit? Because they're kind of like us, right? Not quite, but kind of like us. But if you look at inflammatory bowel disease incidence, at the beginning of the 20th century, the incidence was thought to be about 1 in 10,000 individuals in the Western countries. Now it's 1 in 250. It's a little bit of a change, right? And similar data exists with asthma, hay fever, diabetes mellitus, MS, and right on down the list. And when you look at one of the biggest differences between the Western industrialized countries and the underdeveloped third world countries is they still live in relative filth, right? And if you look at some of the kind of interesting hypotheses that are out there. I'm sure many of you have read about, the hygiene hypothesis, right? That we're too clean now. We don't learn to coexist with all of the things we should be coexisting with. We don't develop good immune tolerance to the organisms we share our planet with that are not really a threat to us. So when we do confront them, we didn't learn to live with them as we got a developing immune system. Eventually, we're going to confront them and we freak out, right? And we get atopic disease and all this kind of other stuff. And this was an interesting editorial on Eat Dirt, the Hygiene Hypothesis, and Allergenic Disease that was in New England Journal of Medicine, talking about that the rise of allergy and asthma and hay fever and all these things, and autoimmune disease in general, has pretty much paralleled the cleaning up of our environment, right? Public health measures and chlorine in the water and public health systems and the use of antibiotics aggressively and all these different things. And that's not necessarily a bad thing, right? There's a yin and yang to everything. There's benefits of all those things that we've done, but there's a price to pay, and there usually is. And it's all these other things. Because when you look, for instance, at the immune systems and the incidence of immune dysregulatory disorders in children that grow up on farms versus children that grow up in cities or even suburbs, it's drastically different. It says your children who grow up on traditional farms are protected from asthma, hay fever, allergenic sensitization, early life contact with livestock, their fodder, consumption of unprocessed cow's milk had been identified as the most effective protective exposures. Studies of the immunobiology of farm living point to activation and modulation of the innate and adaptive immune responses by intense microbial exposures and possibly xenogenic signals delivered before or soon after birth, which means they're exposed to animals and organisms, all kinds of different things that our modern kids growing up in suburbs and urban environments aren't. You see the soccer moms now, right? I mean, these kids live in semi-sterile houses full of carpets and draperies, off-gas and chemicals, but there's not a whole lot of microbes around, right? drive around in the minivans, and then the minute they fall down and get dirty, mom's scrubbing them with triclosan-laden antimicrobial soap, right? I mean, it's a false environment. That's not how humans developed. And they're starting to understand these differences in these different populations. Let me give you another example, interesting. H. pylori, you know, once Robin Warren and Mead and Warren, right, found all this out with the H. pylori, oh, now H. pylori, we got to get rid of H. pylori. It's causing ulcers and gastric cancer and all that stuff. It's bad. Get rid of it. Sort of like the cholesterol, good, bad thing, right? Well, H. pylori is also very, very protective in some ways. We know that H. pylori can cause gastritis and predispose people to gastric cancer if they're susceptible and it's really overgrowing and they can't control it. But it's also linked to protection from allergenic disease, chronic inflammatory disorders, and autoimmune disease. These are my little guys, Ian and Owen. Ian here 7, Owen's 5.5, and this is how they spend most of their days. Not on video games, not watching TVs. They're out in the backyard. We have a creek, we live on a lake. They're catching crayfish, tadpoles, you know, there's chickens over in the neighbor's yard. I mean, I don't have to give them little bottles of soil-based organisms, right? In fact, most times they come to the back slider and knock on it, and I go over and I have to like hose them off to find out it's actually my kids before I let them in the house. They're that dirty, you know. And that's a good thing, you know, they're sampling their environment. And by the way, they're not sick very much, you know, and they're not racked with the same kind of allergies and things that their kids, their classmates and things are. This is interesting. On parasites in your gut actually help protect you from allergies. It's been shown in a couple different models. For instance, there was some really cool studies done in Vietnam and Cambodia and some of those countries on, you know, the kids there just have no asthma, no hay fever, no atopic disorders, none of that. But they're riddled with parasites. They found out when they went in and started trying to treat them as a humane effort for these parasitic infections, all of a sudden they started having all of these different disorders that Western kids had. So started putting these connections together. And the idea is, as we've gotten cleaner and as we've eradicated more and more of these parasites, these helminths, these worm-like organisms, we've seen this rise in these autoimmune dysregulatory conditions. These sophisticated organisms, these helminths or worms, they actually have a metabolism which expresses all kinds of different messengers to our mucosal immune system, which essentially dampens our mucosal immune response. So it suppresses it in a way to allow them to survive, right? What do you think the human response has been over time to these organisms? To dial back up the aggressiveness of our immune response at the mucosal level, to kind of find some homeostasis, right, that we can all get along. And it seemed to have found it for a long, long time. Then we come along with modern health stuff, public health measures, and eradicate all these helminths out of the picture. Now, the immune response, mucosal immune set point, if you will, is set aggressive, thinking that there's going to be this suppressive counteracting force of the helminths that we just got rid of. So what happens? We're in a state of aggression, right? We're in hypervigilance immunologically against our world and against us. And we have autoimmune disease. So this was put into play by Joel Weinstock, who's a gastroenterologist researcher. He did most of his work at the University of Iowa initially, and now he's at Tufts. But he took inflammatory bowel disease patients. And he wanted to test this theory. So he found a helminth that was a good therapeutic agent because it doesn't live, it's not viable in humans for very long if you stop giving it. So if you worry about, you want to give someone some parasites to see if it helps their condition. Now you infest them with parasites, you got to, try to get rid of it later. If you give them Trichoris suis or porcine whipworm, it has all of the kind of immune modulation that you would get from other helminths, but it won't last in the human GI tract for very long if you stop administering it. So when Weinstock did his studies, he did a whole series of them actually, and other investigative teams have replicated some of this as well. When they induced helminth infection into people with inflammatory bowel disease, they induced favorable regulatory T cell changes. They altered innate immunity favorably. So the cytokine patterns and expressions went in the favorable direction away from inflammation and toward pain to anti-inflammatory cytokine expression. You also had changes in intestinal flora which were beneficial. It tended to be good for what we consider the good guys and suppressive of the opportunistic and even pathogenic organisms. And one of the biggest things is it blocked effector T cell proliferation, which is a big part of fueling the autoimmune process. So yes, they actually cured inflammatory bowel disease with helminth agents or parasites. And in fact, this is not, groundbreaking stuff. This is a drug called Ovamed, which is approved in the EU, and it's a helminth-based drug for autoimmune disease and inflammatory bowel disease in particular. And I know that Weinstock, I think, is working with an interest to try to get FDA approval for a similar agent in the United States, but I don't believe that there's approval now. Yeah, I know this is the land of sushi out here. I remember when sushi first started. I'm old enough to remember when sushi really first hit the East Coast, and pretty funny. You'd hear comments like, "Oh, what a nice little garnish on that little pickled ginger and the little wasabi and all." They'd eat the sushi and leave all of that stuff, like it was some parsley on the side of a dish, And the waiter would laugh when he took it away. Oh, those stupid people, they don't understand, right? We know why that stuff's there, right? The ginger and the wasabi and or green food-colored horseradish, which sometimes is what they say is wasabi. Anyway, it's all anti-parasitic, right? So it's the medicine for the parasites you just ate. When we look at the healthy villi of the small intestine, here's a really nice, highly magnified look at the GI mucosa. You can see all these nooks and crannies and all this surface area of all these healthy villi. And in a little capsule biopsy here, you can see all those villi in a normal tissue specimen, and here is someone with inflammatory bowel disease. It's friable, it's like tissue paper, it's not in good shape. So instead of having nice, healthy, one cell lining mucosal layer thick with nice tight junctions, we get a diseased one that's inflamed and has these gaps in between, right? And that allows larger antigenic things to come through. That could be food proteins that are not digested and broken down into small enough amino acid sequences, and they remain in a highly antigenic form. They translate through before they're ready, and you have to react to them immunologically. So sometimes a lot of these food sensitivities or problems you have with tolerating food is not a hardwired genetic fixed thing that can't change. If you take the heat off by not exposing yourself to that food for a while you fix the gut, get the gut, tight junctions tight again, get better digestion, get them off acid-reducing medications, get them on maybe even some pancreatic enzymes, depending on the case. A lot of times you can get rid of these things because this leaky gut, this hyperpermeability, can really fuel the whole thing. It sort of creates this feed-forward type of mechanism. The leaky gut causes more antigenic load, causes more immunological activation, causes more inflammation, which causes more leaky gut, which causes more food intolerances, and it just goes forward and forward and forward until you short-circuit it. And if you look at here at even just the stool analysis, you can see sometimes some objective evidence that there's likely leaky gut, things like lactoferrin being increased, or calprotectin, or white blood cells being present. All those kind of things is not good news. It tells you that there's likely some level of inflammation and or infection going on, or both. And I'm not going to talk a lot about Dr. Fasano's work because he can do that much better than I, but this is what he's really brought to the fore. I mean, we've been talking about leaky gut and hyperpermeability and its importance in chronic disease and things like that in functional medicine for 25 to 30 years. It is not new for us. It's not. But whenever it would be talked about in more conventional circles, it was sort of like, that's that crazy, weird alternative medicine stuff. Well, Dr. Fasano, in his brilliance, realized that this is really a serious component, one of the linchpins of things you need to have autoimmunity be present. And, with his methodical way of going about it and his publications and his research and all of that, he's brought it really to more of the orthodox mainstream medical community in immunology circles and rheumatology circles and gastroenterology circles as being something viable, that this leaky gut thing really has real merit to it. On the other hand, it's been blamed for everything under the sun, so it's kind of the truth is probably somewhere in the middle. But he'll talk about a lot of his work and the genesis of it, and it's really an interesting take. But the only thing I really want to reference here is he'll talk about this three-legged stool of autoimmunity, or this triangle, that you have to have three elements there or the disease won't stand. Okay? One of the things is you need the environmental trigger. We've talked about it, a lot of environmental triggers this morning. Microbes, whether they're bacteria, whether they're viruses, or what have you. Food triggers, food peptides. So environmental triggers in general. You need the trigger. You need the environmental antigen being exposed to an individual that has genetic predisposition to be reactive to it. And what does that usually take the form of? A HLA, or human leukocyte antigen, presenting pattern where they take that specific antigen, environmental antigen, and they grab it and they hold it out like this to the rest of the immune system, to the naive immune system, and go, look at this. This is not me. This is bad, right? Where another individual can be exposed to that environmental antigen and they don't have the right HLA structure to grab that antigen and hold it out and present it. Okay? So they're somewhat immune to it. They don't have this violent immune reaction that someone with that genetic predisposition has. So we have the environmental trigger, the genetic predisposition, and then finally you need the hyperpermeability so that environmental trigger can get through the GI mucosa and have it be presented. So whatever the cause for the leaky gut, it needs to be addressed. And a lot of Dr. Fasano's work involves something called zonulin that he'll explain that can be overproduced in individuals that have autoimmune disease. And zonulin, basically produced by the enterocytes, can hit a zonulin receptor and open up channels or gaps in between the enterocytes and basically create temporary leaky gut. And there's some reasons why that would happen that are advantageous, but if it becomes a runaway train and you have overproduction of zonulin, it's not healthy. And he'll talk about, working toward coming up with zonulin modulators, whether it's zonulin receptor antagonist, whether it's other things that modulate zonulin through other mechanisms. But it is definitely a protein that does modulate intestinal permeability, and it has some other functions in the body with as a haptoglobin, and I'll let him discuss that. We've used a lot of stuff in functional nutritional medicine to heal leaky gut for a long, long time. Things like L-glutamine, of course. L-glutamine is the preferred energy or fuel source for the enterocytes, and if they're deprived of good levels of L-glutamine, they atrophy. They learn this very quickly when they put patients on tube feeding or enteral feeding and or IV feeding, I should say, and if they don't get glutamine down into the GI tract, their GI mucosa sloughs off. So this was found out in critical care medicine very early on. But L-glutamine is very, very important for the GI cells, of course. But there are a lot of other things that have long traditional use in medicine, herbal medicine, even modern naturopathic medicine, botanicals that are mucilaginous, that coat the GI lining and help it heal up from inflammation. These are things that are classically anti-ulcerative, and they're usually slippery kind of things, right? So slippery elm, okra, if you ever cooked okra, it has all that sliminess to it, right? That's the mucin, that's the properties that can heal the GI mucosal lining. Things like aloe, marshmallow, right? Marshmallow, they're not those little cornstarch puffs that you get in the store. The traditional, origin of those is the marshmallow plant. It's a mallow family plant, grows in wet, marshy environments, so it's a marshmallow. If you pick it up by the stem of the root, it comes out with these little white bulbous things that are underground that look like marshmallows that you buy in the store, but they're not. They're actually real, right? They would snip the roots off each end, put it on a stick, and crisp it up in a fire and eat it. And it had medicinal properties. It healed the gut. So they simulated it with cornstarch puffs. I don't know what to say. And they couldn't call them marshmallows legally originally, so they called them marshmallows, right? And now they actually call them marshmallows too. But cat's claw. So there's a lot of great botanicals, but pepsin GI, which is zinc carnosine, it's a complex of zinc and carnosine, very protective to the GI mucosa, very anti-H. pylori. We use it in H. pylori therapy all the time, but it's just really good to heal up the gut lining. It's used in Europe all the time. It's co-administered as a drug with agents that are very caustic to the GI tract. So it's used concomitantly with things like indomethacin and biphosphonates and things like that, which really tear up the gut. And then something I want to spend a little bit of time talking about, which is N-acetylglucosamine, which is really a fascinating thing. N-acetylglucosamine, this is a study, it says it shows promise as an inexpensive, non-toxic treatment in chronic inflammatory bowel disease with an action that's distinct from conventional treatments. It's potentially helpful in stricturing disease. What N-acetylglucosamine, so it's a glycan, right, a proteoglycan. So N-acetylglucosamine is something that's a basically sugar protein together. It attaches to T cell receptors and forms branches or lattices on the cell surface that prevent the receptors from clustering with near where antigens are located, and you get less clustering, less TH. It says less TH activation of Th1 cells and less autoimmune proliferation. So what you have here is if you have an antigen-presenting cell with the antigen, and then you have a T cell that's looking to try to dock to this and cause a autoimmune reaction or proliferate an immune response, if you give N-acetylglucosamine, it kind of fills in these gaps here so that you can't get binding here. You don't get a docking, essentially. It's just like when everybody knows about cranberry and UTIs. Why does cranberry work? It has a sugar in it called D-mannose, which is structurally similar, that binds on the receptor site so the organism can't form a locus of infection. It can't bind to the bladder wall, right? So these blocking agents, these glycobiological blocking agents are very, very effective at fighting certain types of infections and immune responses. So N-acetylglucosamine is really a good, interesting agent. This is a paper by a friend and colleague of mine who actually spoke at this event when we did it in Florida last time, Jerry Mullen from Hopkins, looking at a lot of the different natural agents that are very helpful in immunomodulation, everything from resveratrol, green tea, curcumin, boswellia, all these very highly bioflavonoid-laden botanicals, fish oils or omega-3 fatty acids, vitamin D, and probiotics. So you can pull that paper. And vitamin D is very interesting. A lot of people think of vitamin D and autoimmunity. Well, it's an immunomodulator, so it's good. But actually, vitamin D has a whole long list of mechanisms by which it could be helpful in autoimmunity. One of which is that, look at this. In vitro experiments demonstrate that the VDR, the vitamin D receptor, mediates the activity of 1,25-hydroxy D3 that induces junction protein expression and strengthens the tight junction complex. So vitamin D directly treats leaky gut. A lot of people don't know that, right? It's not just an immune modulator, it's actually frontline therapy for leaky gut. But of course, vitamin D has been shown to work on at least 229 genes, some of which have been associated with Crohn's and type 1 diabetes and all kinds of inflammatory things. Genomic research has found almost 3,000 binding sites where vitamin D attaches to the genome, so it's a very complicated picture. But one of the things that vitamin D does is more activation of vitamin D by conversion from 25 to 125 form increases your endogenous production of something called human cathelicidin antimicrobial peptide, or CAMP. And what CAMP is endogenous antimicrobial peptide. It's your onboard antibiotics, right? Like Roundup Ready soy or whatever. Your antibiotic ready humans, right? You make your own antimicrobial peptides, and vitamin D is the signaling molecule to do this, or substance to do this. So as your vitamin 125D goes up, your cathelicidin or CAMP goes up, and this is an antimicrobial. Well, how would that help in autoimmunity? Let's say MS. Well, vitamin D is supposed to be great for MS. How is that helping MS? Well, if it's inducing the production of antimicrobials, this is Charles Stratton's work at Vanderbilt, and he shows that MS patients harbor stealth infections with chlamydophilia. Maybe that's the molecular mimicry environmental agent in some patients with MS. Because here's some other work on infectious causes of multiple sclerosis with viruses as the triggers. So you see, it's a complicated story, right? It's not just one agent, but there is reason now that we understand why when people put on antibiotics, let's say, for other issues, other infections, their autoimmune conditions tend to get better. This is also really interesting work by Pender looking at reactivation of Epstein-Barr virus, chronic Epstein-Barr infection in patients that are genetically CD8 T cell deficient. That if you have this kind of unhappy triad of CD8 T cell deficiency, and it gets worse as you get older, Epstein-Barr virus infection, chronic stealth infection, and vitamin D deficiency, he gives a really cool progression toward autoimmunity that can occur. So once again, another kind of variation on the theme. But to wrap up here, because my time's drawing short, where are we going with autoimmune disease? This is a really interesting piece that occurred in Scientific American on predicting autoimmune disease. New predictors of disease, molecules called predictive autoantibodies, appear in blood years before people show the actual clinical symptoms of the disease. And these tests to detect these molecules could warn of the need to take preventive action. And this is actually out of New England Journal of Medicine showing that you don't go from normal immunity to clinical illness with an autoimmune disorder, let's say lupus or MS or whatever, just like that overnight. There's some exceptions. Type 1 diabetes happens really quick. Graves or Hashimoto's can happen pretty quick. But generally, you go through a progression where you're exposed to environmental factors, you have genetic influences at play, but you go from normal immunity to benign autoimmunity, whatever that means. You have autoimmune reaction, but you don't have clinical expression of the symptoms yet. Pathogenic autoimmunity, and this has to get bad enough before some doctor queues up and says, well, you have this symptom, this sign, this lab test, you have X, right? When they actually diagnose a disease. And if you look at this progression, this is on a model with lupus, you can see the predictive autoantibodies in the blood of lupus people generally at least 6 years before clinical diagnosis with lupus is possible. So, if these antibodies appear in blood years before the presentation of symptoms, could they be used as biomarkers? Can we use them preventatively, not after the horse has left the barn, but before it's left the barn, right? Look, these are some of the predictive autoantibodies in diabetes, for instance. But then, if you look at some of these are slides from Tom O'Brien, who compiled all of this from the literature, but if you look at the disease, the autoantibodies, the PPV, which is positive predictive value, and the years before clinical diagnosis that you can find these things, it's striking. If you look at some of the antithyroid peroxidase antibodies and things in Hashimoto's, if you find them, it's got a 92% positive predictive value that that patient will go on to develop Hashimoto's within 7 to 10 years, for instance. If we look at Crohn's, your anti-Saccharomyces cerevisiae antibodies, almost 100%, and within 3 years. Here's the celiac, anti-tissue transglutaminase, anti-endomysial antibodies, the classic stuff, the HLA, DQ2, DQ8, highly predictive within 7 years, generally. RA, rheumatoid factor, anti-cyclic citrullonated peptide, pretty high positive predictive values up to 14 years before. Well, what if we were using these things more in a screening methodology, right? If they became arrayed in large panels and they were very cost-effective to do, and we can do them as part of our normal workup. I mean, we're wellness doctors, right? We do preventive medicine. Do we really? Sometimes we say we do, and I'm not sure how much we do, right? But these are some leverage tools to really do it, particularly in patients who we know, older people in their family, have inflammatory arthritis, have autoimmune thyroiditis, have MS, have developed early dementia, whatever it may be. We can intervene with things like this. Look at these positive predictive autoantibodies, and if we find them, we know their train is heading down the track to that station of that autoimmune disease. We need to pull the track apart and not let it get there. How do we do it? We stop these dominoes from falling by removing all the environmental triggers we can, finding the foods they react to, cleaning up their GI environment, getting rid of any of these microbial viral associations that we can, that we at least know of now, treating the leaky gut, making them less inflammatory, getting them on a cleaner diet and lifestyle, and maybe, just maybe, we can stop the progression to that disease. And labs are now building these arrays. I mean, all these these predictive antibodies are available through standard commercial labs, generally, you know, Quest and LabCorp, but they're a la carte, right? Or they're bundled up in more smaller panels, like a rheumatoid panel, right? Or a celiac panel. But we're talking about arrays that cover a whole wider spectrum of these things, and you can run a patient through it and say, let's say, you know, these are all these predictive autoantibodies across an array of systems and organs, and this is a healthy patient's response. And then you can actually come up with sort of actuarial risk data for these different diseases, if you'd like, versus someone who hits on a bunch of these different things, or even hits very strongly in one system related to one disease. You can have a much different risk assessment for them on will they progress. And then looking at dietary proteins, looking at some of these cellular response tests, food tests we talked about, infections by looking either at, you know, viral titers, lymphocyte subsets, looking at molecular stool analysis, whatever it will take, depending on what you find in the positive predictive antibodies. And even, I didn't get to talk much about chemicals and toxins. There are a lot of toxins, particularly the metals. The metals, the heavy metals, are transitional metals on the element table. They will bind to the sulfur groups of biological proteins and create haptens, which are immunologically active. They will induce an autoimmune response because it's taking your host proteins and making them foreign by adding that metal on the end of it. Now it's a foreign protein, and boom, you react to it. So that's the basis of like the ELISA test, the immune response test, the mercury. So I didn't really have time to talk about that, but that's an important role. So assessment and intervention for autoimmune disease. Detect and remove opportunistic pathogenic GI bugs. Detect and eliminate food sensitivities. Predictive autoantibody testing, employ that if possible. Check for toxins and support good, healthy detoxification. Check vitamin D status and optimize that. I didn't get to talk about some of the vitamin D controversies with Marshall's work and activation, hyperactivation of monocytes with stealth infections, converting 25 to 125, making it look like there's a deficiency of 25, and treating with pulse antibiotics and angiotensin II receptor modulators. It's really interesting stuff. I just don't have time to go there, but look up some of Marshall's work. It flies in the opposite direction of most of the vitamin D experts' opinions on vitamin D and autoimmunity, like Hollick and Cantora and Cannell and all of those folks. And I think they both might be right, just in different situations. Most of what Marshall was talking about seems to be in play in sarcoid and a few other disorders. Quench excessive inflammation and oxidative stress. A lot of that has to do with their modulating their diet, right? Nutritional interventions, anti-inflammatory diets, low arachidonic acid, getting rid of the highly potentially antigenic things, just as a matter of course, such as like gluten-containing grains and potentially dairy. And gastrointestinal restoration, really making the gut as healthy as it can be. The GI mucosa, the mucosal immune response, the GI microbiota, and stress reduction. That's one I also didn't get a lot of time to spend on, but stress is really pulling down our immune system. You give too many corticosteroids, what happens to your immune response? Down, right? One of the first things that happens is you reduce your production of secretory IgA on mucosal linings. You reduce secretory IgA, you get leaky gut, you get antigen penetration. So can stress cause food sensitivity? Yes, it can. There's a pathway there for that to happen. Can stress be involved in dysregulating the immune system and leading to autoimmunity? Sure it can. Pumping out too much cortisol, too many catecholamines, it's not a great thing over long periods of time. These are just a couple of book references I'd recommend that I was author in, but integrative gastroenterology really picks apart a lot of this stuff I was talking about from gastroenterology standpoint, and some of the testing, particularly the molecular-based stool analysis testing and things like that, functional evaluation for integrative and functional medicine, and then foods and nutrients in disease management by Kolstad. I go into, in my chapter in there, I go into some of these same things. And just in closing, I have some information in the back at the ALCAD booth on the master's program in clinical nutrition, human nutrition, and functional medicine at the University of Bridgeport, where I chair that, or I direct that department. So if you want greater depth of exploration of some of these topics, that's a place, and as well as the MS program through A4M as well. And otherwise, just thank a couple of people for borrowing some graphics and for them educating me along the way, including some of the speakers here, Dr. Perlmutter, Dr. Fasano, Dr. Davis, and others. So thank you, everybody. Appreciate it.